Hello and welcome. We are at the so-called home run. This is the penultimate lecture of the last module, and we are at the end of our course on Nanobiophotonics. I do feel slightly melancholic, but at the same time I feel good that a substantial amount of knowledge I was able to transfer to you, spanning from biology to physics to medicine to brains to optics and photonics and whatnot. So, this is the penultimate lecture. So, let us talk about biotemplates. I told you in this topic that we utilize biological materials, naturally existing nature created biological materials, as some sort of a mold, as some sort of a template, and then put some sort of polymers or nanoparticles in the specific sort of arrangement that this template allows us to do, right. So, what exactly are those biotemplates? So, a biotemplate approach is a top down. By this time you should know what is top down versus bottom up approach in nanotechnology. It is a top down method used to create nanostructured photonic materials. It obviously draws inspiration from natural microstructures. They are natural microstructure. These can be utilized in their original form or modified version. The approach enables the development of assembling and processing techniques. The biotemplate approach allows cooperative amplifications, and there is wide range of biological materials have a wide variety, they have wide distribution. Even in a small 1 micron by 1 micron area of a virus or a DNA or a bacteria, you will see several types of features, several types of nanostructures. If we use it as a mold and try to create a negative structure, by putting some sort of a polymer. So, suppose you have a viral structure like this, with these being the spike proteins, with this is the core spike proteins. You deposit some kind of a glue, some kind of a polymer in between this gap, and then somehow you remove this virus. You either dissolve it in some organic compound or somehow you remove it. So, whatever remains, whatever that polymer remains will be a negative structure of the spherical virus with spikes in between. So, it will be an inward structure with lots of holes. Instead of spikes, instead of cylinders, you will have lots of gaps, lots of dots, lots of hole in between with a hollow core, So, imagine the coronavirus particle that you have seen all over, a negative structure of it, So, two very important biological materials that we can utilize as a biotemplate. Number one is DNA. One example of biotemplate is DNA. DNA structures can be used as templates to synthesize nanostructured photonic materials, allowing precise control over their optical properties. And the second being virus, as I was saying. Virus can be utilized to guide the assembly of nanoparticles with tailored photonic properties. So, having that kind of a negative virus structure with holes and hollow cylinder and whatnot, you are thereby periodically varying the refractive index. You are thereby both in virus as well as in DNA, you are periodically varying the refractive index with polymer air, and thereby restricting the movement of the photons, and in essence making some kind of a optically active electromagnetic structure, or electromagnetically, let me rephrase it, electromagnetically active nanostructure. Can you make picostructure? With different strands of DNA, what would be the length of one single DNA? Is it in angstrom? Is it in picometer? So, instead of having nanotechnology, are we talking about angstrom technology? I recently had the pleasure of attending a lecture of Professor Nikolai Zeludev of University of Southampton, and he was talking about pico optics, not nano optics, and these were some of the ideas that he provided. So, let's try to discuss a little bit more about deoxyribonucleic acid, DNA, as a smart glue. So, as I was mentioning, in photonics as well as in nanotechnology, DNA molecules are used as biotemplates to guide the assembly and organization of nanoscale materials into specific structure. You know that DNA has 4 nucleotide bases, A, C, T, G, and they are all paired, A with respect to C and G with respect to T. So, if you have C here, you almost always able to say the complementary pair being G. On the other hand, if there is T here, you know that the complementary pair will be A. There is no other process, there is no other way. Well, couple of exceptions always exist, but 99.9999% case, if you know one set of bases, one particular sequence, you can immediately calculate the other sequence. So, nanoparticles or other nanoscale materials such as quantum dots, metallic nanoparticles or semiconductor materials are functionalized with complementary DNA sequences. So, you can put your negative structure or you can put your polymer or quantum dots either in the gap region, make some sort of a negative ladder structure, consider it as a ladder. In between the space you have put some sort of a nanoparticle, polymer, nanopolymer, quantum dot and whatnot, or you can put it alongside in certain of the bases and then try to create another complementary pair and combine them together and later remove it, so that you have a negative structure. These white spaces where I am having my cursor, the white the laser pointer, you can have some kind of a nano structure with this precise location. And if you are clever enough so that you have made some kind of a quantum dot, a specialized quantum dot, specialized nanoparticle, which will only attach with C, which will only attach with T, which will only attach with G and A, then by this you will have a complementary nanoparticle based periodic structure with particle A, B, C, D and so on and so forth in that arrangement. So, nanoparticles or other nanoscale materials are functionalized with complementary DNA sequences. When mixed with the complementary DNA sequence on the template, the nanoparticles assemble along the DNA strands through base pairing. So, through base pairing, C matches with G, you have created a nanoparticle X that is specific to C and nanoparticle with Y specific to G. Now, you have arranged them together, you have arranged a particular one together and have tried to mate C and G. By definition, X and Y will also mate, right. So, if the refractive index of X is say 2 and the refractive index of Y is 2.5, you will have 2, 2.5, 2, 2.5, 2, 2.5 or mix and match. So, all of these things is available where we utilize DNA as a template, and imagine the resolution, imagine the how small it is going to be. So, nanotechnology probably is passé now. Now, you have to look into pico technology or angstrom technology. So, as I was saying, you can have this gold nanoparticles complementary strands that can either be attached with the base pairs or you can have it with the phosphate base, the backbone. So, the self assembly process along the DNA template leads to formation of specific structures dictated by the design sequence. So, this allows you this controls it so that the nanoparticle are arranged in a specific pattern. A particular pattern is maintained. This could include linear chains, periodic arrays and even more complex 3-dimensional nanostructures. The arrangement of nanoparticles along the DNA template results in unique optical properties due to plasmonic interaction, quantum confinement, whatnot. So, there will be a combined effect between them. They will have individual plasmonic resonances. Plasmonic resonances is because of the localized electrons. I told you the electron that comes to oppose incoming light radiation, but if you have arranged the plasmonic nanostructures in such a way with each nanoparticle identical, exactly same as the other one, then the localized surface plasmon resonance, the electron cloud here versus electron cloud here versus the electron cloud here are all same, because they are identical particles. And when you have the same electron cloud combining together, they will talk and they will resonate, and the result, the output result is creation of a huge, LSPR signal, resonance surface plasmon, localized resonance surface plasmon signal, that could be utilized as a sensor, because these signals that are generated by the free electron present in these gold nanoparticles, they are very, very susceptible to any change in the local refractive index. So, you just cover one of them with some amount of a molecule, a biomolecule, something is just attaching it, the entire structure will get disturbed, the resonance of the entire structure will get disturbed, because previously they were just identical to one another. Now, one of them is non-identical, i.e., you have introduced defect, So, there is a small red shift, and that could be utilized to detect the presence or absence of any particular molecular species, meaning you are going into so called single molecule detection. So, the arrangement of nanoparticles along the DNA template results in unique optical properties due to plasmonic interaction, quantum confinement. This makes these structures highly tunable for various photonic application, and by controlling the sequence of DNA templates and the type of nanoparticle used, researchers can create functional nanomaterials with specific optical responses that can be used, as I said, as sensors, optical switches and other photonic devices. Similarly, you can have virus as a template. So, this is a typical example of a virus. Now, see how these holes inside this along with this capsid, along with this viral body is arranged. Now, imagine a negative structure, i.e., the holes you will fill it with some sort of a polymer, some sort of a nanoparticle, and the rest as it is, and then the nanoparticle is removed. So, what kind of a unique photonic nanostructure you will get by having nanoparticle arranged in this particular pattern? Please do not think that they are random, these holes, this pattern, this yellowish versus greenish, these are not randomly arranged, just like you see the colors in butterfly wing or in peacock feather, they are not randomly arranged. Similarly, these holes, this crest and trough along the body of the virus is not randomly arranged, they have a particular purpose. Combinedly, individually you may not find a particular purpose, but when you combine them together, probably this makes this thermodynamically stable, probably it helps it to protect the nuclear material that is inside, the RNA or DNA material which is holding inside, or probably it has some kind of a spike proteins associated with it, which can be used to attack a particular cellular receptor somewhere in a particular cell and thereby open it up. It is just like lock and key mechanism, I told you, it is just like lock and key mechanism. Those spike proteins acts as key, those things that you saw in the coronavirus, they act as a key which attaches with a specific opening of the semi-porous cell membrane, opens it up and inserts the DNA or RNA presents inside the virus into the nucleus, through the cell, through the cytoplasm into the nucleus of the cell, hijacking its reproductive property, reproductive machinery, and the cell reproductor machinery is no longer utilizing to create copies of the cell, but the cell reproductory machinery is being used to produce copies of the virus instead. So, the first process comes when the cell membrane's particular door, particular lock is opened by a specific key, and these spikes, more so in a coronavirus, you have seen that red color spikes are keys, right. So, viruses possess well defined morphology, flexible microstructure and microdiaphyable surfaces, making them suitable template for creating innovative photonic materials. Copia mosaic virus CPMV particles, which are 30 nanometer diameter, can serve as template for attaching various components to their surface. Mutant CPMB particles are functionalized, altering their properties to suit specific application, and using a dimethylamine reagent, functional CPMB particles are reacted with dye molecules, and you can utilize it to attach gold nanoclusters onto the surface of CPMB, and thereby you can create some kind of a resonating plasmonic nanostructure that will have their own effect. So, instead of designing some sort of a sensor using nanoparticles from your own imagination, just look into nature. Just look into nature, they have a huge variety, just look into them, and if you can copy or mimic that, utilize those design already existing, because nature has got like few billion years to do permutation combination, and finally it has come to the most efficient design. You know about evolution, you know about survival of the fittest, natural selection. So, the final product after 1 billion years of evolution is something that is most efficient, right. So, instead of you trying to make trial and error designs of nanoparticles which may or may not work, why don't you look into already existing design present in the nature, which nature did over a period, nature experimented over a period of 1 billion years since life form existed in this planet, and it has already have come to the fittest, the most efficient, the best result. Simply copy it, right. So, this approach enables the creation of regions within a significantly increased concentration of attached species. By attaching specific components to the virus template, localized optical properties can be achieved, and the use of virus template utilized for tailored optical response and functionalities. So, this is a very cutting edge technique. These are hot topic of research. You keep on asking, can you give me some hot topic? I see it in the forum. Can you give me some hot topic for research in biophotonics, nanobiphotonics? So, this is it. Utilize your viral particle as some sort of a template and create unique photonic nanostructure, electromagnetically active nanostructure, even picostructure if you so can, and see how it is able to localize light, diffract light in a unprecedented manner. Who knows what new phenomena of light you might come up. Negative refraction is already there, switchable negative refraction. There is something called slow light, where the group velocity of the light is tremendously reduced. You should know what is the difference between group velocity and phase velocity by now. So, obviously, this is something of a very hot topic. At the same time, you can utilize bacteria as a biosynthesizer for photonic polymers. Pseudomonas bacteria produces a family of these alkanoic acid polymers, thermopolymer for example. This alkanoic acids pHs typically have a molecular mass from 50,000 to 100,000 Daltons. Da is Dalton. This is the same Dalton that gave the idea of atom, something that cannot be divided, atom cannot, indivisible, but now even a school child knows how atoms can be divided into what not. So, bacteria synthesized PHA polymers have been fabricated into thin films. Chromophores such as APSs can be successfully doped into this polymer, making fluorescent material. So, the output, the byproduct of bacterial reaction, bacterial mating, bacterial reproduction is this thermopolymer, a polymer which reacts differently with respect to different temperature being put into it, and thereby you can try to see some kind of a thermo-optic effect, where the refractive index change of a particular material because based on the temperature, and if you have divided the temperature gradient somehow, you can change the refractive index and thereby change how light gets localized. Sometimes opposite is also true. By photothermal heating, by absorbing light at specific areas of that polymer, localized heating can be achieved, and then that localized heating, hot cold hot cold hot cold periodicity can be utilized for several different purposes, something that I use for making sensors. Certain species will have an affiliation for a particular temperature, certain chemical molecule will have an affiliation towards a particular temperature. Every chemical reaction has an optimum temperature. So, the particular chemical species, particular virus will like to go to a specific temperature area, and by that, if the refractive index changes, we can also utilize it for the detection. So, what is the future? We go for nanoscale template and self assembly, we go for biomaterial based self assembly, we have multifunctional structure, we have control of photo processes, and obviously holographic data storage. So, that brings me to the conclusion. Biomaterial provides a unique platform for photonic applications. Nucleotide base pair interaction in biological system offers precise control, and mutation techniques enable the creation of variant and tunable structures that can be utilized for several different applications. So, these are the concepts that I tried to cover for today, and these were my reference. I will see you in the last class. Thank you.